everyone. We are now starting the session in Music Box. So this here is Rohit, and he'll be talking about integrating biological knowledge graphs with multi-modal biomolecular data using GNNs. The structure of this is that it is 35 minutes of talk and 5 minutes for questions at the end. And so I will now let Rohit get started.
Thank you very much, and yeah, thank you to everybody who's in this room right now. My name is Rohit, as Mars just introduced me. I work at the Jackson Laboratory for Genomic Medicine. We are a non-profit biomedical research organization working on understanding the genetic molecular basis of complex diseases and using that to develop therapeutics for challenging diseases, and this talk will be some of the work that I have been doing. Me and my team, we've been working specifically for Alzheimer's disease. I work in the ML and imaging team at the Jackson Laboratory, and this is one of our projects. Essentially, when you think of it as an ML case study, this is essentially a multi-modal data fusion problem. So we'll just get started. So just a quick background about me. Like I said, I am a computational scientist at the Jackson Laboratory for Genomic Medicine. We're based in Farmington, Connecticut. We work on a number of different biomedical data science problems, including multi-modal data fusion, such as multi-omics data integration, also translational research between model organisms and humans. I personally work on fundamental computational problems such as the interpretability of graph neural networks. We also look into drug discovery and variant effect predictions and on various types of biomedical problems. Prior to my appointment at the Jackson Laboratory, I did my PhD from Purdue, where I worked on some ML problems in physics, applying them to partial differential equations. I was in an engineering department, but did a lot of applied mathematics work. And after my PhD, I transitioned into computational biology. I went to the Wolfgang Harbor Laboratory, which is another research institution based on Long Island, where I did work at the intersection of deep learning and genomics. Okay. So, I'm going to be talking about Alzheimer's. Before I talk about Alzheimer's disease, specifically, I want to offer a little bit of historical context. Through the history of genetics, at least in the early history of the field of genetics, I would say in the first half of the 20th century, it was the view of geneticists that diseases were caused by individual genes. And while there might have been some, there was this view that there could be some traits or diseases that could be caused that could be polygenic. By and large, the prevailing view was that traits and diseases in general were caused by singular genes. The difficulty with exploring this in the early history of the discipline of genetics was essentially technology. It's a lot like how deep learning didn't really explode until the 2010s, where compute became readily available. And so a lot like that with the development of genetics and genomics. As the field has developed through the second half of the 20th century and the 21st century, what we've come to understand is that diseases in general, far from being caused by singular genes, are highly polygenic. I just want a quick temperature check. How many here have some background in some formal background in biology? Okay, a few. So yeah, we've come to understand that diseases are highly polygenic, which is to say that they're caused, at least their incidence and severity is a function of the interactions of many, many different genes. And in fact, particularly in the 21st century, in the last couple of decades, as biotechnological advancements have taken place, what we've been able to do is that we've understood that it's not, in fact, it's not just an interaction between genes, it's an interaction across multiple molecular layers, across multiple modes of molecular interactions. Genes are specific regions in the human genome, but they constitute approximately only 2% of the human genome. It used to be that we had 98%, the remaining 98% of the human genome was considered junk DNA. And we've come to now understand that they're far from junk DNA. They, in fact, there are specific regions of what we call the non-coding DNA that interact with genes and affect the functioning and the regulation of genes. And it's not just DNA, it's also proteins, metabolites, lipids, fats, and so on. There are epigenetic markers that influence the functioning of genes. And so this is essentially a case study to understand Alzheimer's from a multi-omics perspective. When you hear the term omics, how many of you are familiar with the term omics? A few. And when you hear the term omics, basically what you should think of is a molecular modality. It could be genes, it could be proteins, it could be metabolites, it could be methylation sites. But what you should think of is one molecular mode. And multi-omics is several molecular modes. So this work is essentially an attempt to extend our understanding of Alzheimer's and to identify potentially some new therapeutic targets for Alzheimer's by conducting an analysis of multi-omics data for Alzheimer's. So Alzheimer's specifically is a progressive neurodegenerative disorder. And what that means is that the disease worsens over time. That's what a progressive disorder is. It's neurodegenerative, which is to say that it affects the nervous system. It causes cognitive decline. It causes a decline in your motor skills. And Alzheimer's is far and away the predominant form of dementia. There are other forms of dementia, such as Lewy body dementia, vascular dementia. I'm not sure if you've heard of these terms, but far and away, Alzheimer's is the predominant form of dementia. If you take one biological factoid from this introduction to Alzheimer's, it's this bit of information. Alzheimer's disease is renowned for having these two very specific molecular signatures. One is that it causes the formation, is that you see formations of these plaque-like structures. They're called amyloid beta plaques. Another thing is there's a protein called tau, which undergoes a process called hyperphosphorylation and results in the formation of neurofibrillary tangles, it's a little difficult to pronounce, or NFTs, not that type of NFT, this is different. So these are the two hallmark signatures of Alzheimer's. It doesn't mean, if you see people with these signatures, it's not 100% guarantee that they do suffer from Alzheimer's, but they're the most reliable predictors, pathological predictors of Alzheimer's. And Alzheimer's disease, in fact, has a very, very varied pathology. There are lots of genetic risk variants that are associated with Alzheimer's. There are lots of epigenetic markers that are markers of Alzheimer's. They show up in these signatures, these types of signatures. And when therapeutics are developed, these are the two principal signatures that drug developers will try to attack. So even though we know about the various pathologies that are indicative of Alzheimer's, the reason that Alzheimer's is, if you look at the market for Alzheimer's therapeutics at this point, I'm not entirely sure. So I think there's maybe 3 or 4 drugs that are currently available on the market for therapeutics that are currently available on the market for Alzheimer's, and they're only meant for people with early detection of Alzheimer's with mild symptoms. There's essentially no cure. There is no cure for Alzheimer's to the extent that there are a few therapeutics that are available now for Alzheimer's. They only halt the progression of the disease. And so And so we see cost of care for Alzheimer's spiral year after year after year. It's a big public health crisis at this point, where I didn't have this figure updated until a few days prior to this talk. I looked at this figure recently. This figure is updated for 2023. So as of 2023, the annual expenditure on Alzheimer's disease care has spiraled to $350 billion. And the reason is that, in spite of the fact that we do know all sorts of pathological signatures for Alzheimer's, we don't really understand the molecular mechanisms of the disease. And so, given that it's a big public health crisis, there is now ongoing many different large-scale multi-institutional public-private research efforts that target all sorts of different aspects of AD research. So we try to develop different model organisms to study Alzheimer's. We try to develop therapeutics, look for new drugs, and on and on. Some of these consortiums are called AMP-AD, Model AD, GKD, and there are lots of other similar research efforts. And these are essentially multi-billion dollar, decades-long research efforts, mostly funded by the NIH, but also private funders. Okay, so I'm still here to present an ML case study. That was a little bit of an introduction to the disease itself. So what I would like to do is to tell you about where I would like to begin is to tell you where the data actually comes from, right from the source. We should always care about where our data actually comes from. Where does multi-modal multi-omics data come from? You can profile many different types of biomolecules. Broadly speaking, there's two types of approaches to profile or measure the activity of biomolecules. One of them is through the sequencing of DNA. There are many different types of biomolecules that rely on sequencing technology. The most common technique is what is known as RNA sequencing, or RNA-seq for short. RNA sequencing measures gene expression, which is the activity of genes. And far and away, this is the most popular, the most common type of biomolecule that is profiled for studying diseases or for just generally anything. You can also look for epigenetic patterns. There's something called DNA methylation marker, which is done through a technology called bisulfite sequencing, or BSC. You can also, our DNA wraps around itself. You can look at how tightly coiled your DNA is through a technology called ATAC-seq. It measures how accessible your DNA is to proteins that bind with it to cause gene expression. So these are not directly, so DNA methylation, DNA accessibility are not directly related to genes, but they affect the expression and the regulation of genes. So the other category of molecules that are profiled come from the use of a mass spectrometer. Proteins are the most popular type of biomolecules that are profiled using a mass spectrometer. But you can also profile lipids or fats, profile small molecules, metabolites. But broadly speaking, you have these two categories. Okay, so that's how we get raw data. You can't do ML on raw data. How do we actually get data how do we actually take the raw data, process it, and into a format that we can actually work with and do any kind of analysis with? There's a lengthy bioinformatics pipeline that is associated with every individual type of molecule and every individual type of technology. So for example, there are many different types of proteomics protocols. Every single one of them will have their own bioinformatic processing pipelines, and so on. And all of these steps that are involved in processing raw omics data and to produce it into a format that is analyzable is constantly evolving. For example, with gene expression, the technology used to measure the activity of genes. The key step here is the key bioinformatics step that is used is to map reads from the DNA sequencer to the genome to figure out which genes are actually being expressed. And it's called the reference genome alignment. And there's some quality control checks. But essentially, all of these, no matter what type of molecule, what type of technology, all of these processing pipelines result in a final format of the data, which basically looks like this. It's a table. That's what it is. It's a table. Samples along the rows. Those abbreviations that you see are basically some gene symbols. This may or may not make sense. May not make sense to most of you, but those are just genes. And the numbers, the annotations in the cells are basically telling you how much that particular gene is being expressed or how active it is in that particular sample. That's what it looks like. It's tabular data. And so the fusion problem is just this supervised learning problem. Take all of that data, whatever modalities you have, and learn a map. And what are you mapping into? Whatever target phenotype you're interested in. It could be severity. Typically, this is the severity of a disease. You can make it into a classification problem, regression problem. It depends on how you frame the problem, but that's what the supervised learning problem looks like. You take all of that data, you map it to some target phenotype. In this case, for this talk, it's just going to be a binary classification problem where you're targeting it to the phenotype that you're mapping the data to is whether or not that particular patient has AD or doesn't, given the pattern of expression of their various biomarkers. So what is challenging about this particular type of supervised problem? Well, these are just the standard challenges with multimodal data fusion. This map, F, that takes you from the molecular measurements to the target phenotype is typically nonlinear. Right? It's a complex biological signal, it's nonlinear. Unlike multimodal data fusion problems that you might have worked with when it comes to text or video or those types of modalities, the problem here is that you're working in this high-dimensional space where the number of features that you're looking at is much larger than the number of samples that you have. You have maybe a few hundred patients. You have thousands and thousands of biomarkers individually within each modality, and then to put it all together. So there's interactive effects. There's interactive effects within a modality. There's interactive effects across modalities. Also, you might not have measurements across all modalities for any given sample. Measurements are incomplete. So yeah, I mean, most of these are standard multimodal data fusion problems. Specifically for omics data, the biggest challenge is that it's high dimensional. The number of features is much greater than the number of samples. Okay, so the data. What data do we actually use? Within aging and Alzheimer's, there is a long-running epidemiological longitudinal study known as ROSMAP. It stands for Religious Voters Study Memory Aging Project. And I think it started sometime in the mid-90s. As of now, there are approximately 3,000 participants for whom all kinds of biomedical data has been collected. It's a longitudinal study, so that participants undergo clinical and neuropathological assessments annually. Within the study, within the framework of the study, you have molecular measurements, you have clinical measurements, you have imaging data like MRI, PET scans, and so on. And so researchers within dementia, Alzheimer's, and aging generally essentially consider the ROSMAP study to be the primary benchmark with which to test new methods. This is our main source of large-scale public data. Okay, so we are not going to, we don't look at all of the modes altogether. I'm just going to talk about two modalities, integrating two modalities. We're going to look at gene expression or transcriptomics, and we're going to look at proteomics. Our data set has those many number of samples. You have about 200 samples that are shared. So 200 patients have 200 patients have measurements of both gene expression and proteins. And then you have a lot of patients who have protein only and gene only measurements. And you can see the number of features that you actually have. You have approximately 20,000 genes and about 9,000 proteins. It's an order of magnitude higher than the number of samples that we have. All of these samples come from a specific region of the brain, the dorsolateral prefrontal cortex. Most of the AD measurements within ROSMAP come from that region. Also, although there are other brain regions from which measurements are collected, different brain regions have played different roles in cognitive function and the relationship to AD disease progression. We look at this one tissue. And we do some filtering on this data. Okay. So we have 20,000 genes and we have a few hundred samples, 9,000 proteins, 20,000 genes. When you are faced with a high-dimensional data analysis problem, your first thought is probably do some data analysis, dimensionality reduction, some sort of feature selection. If you're a statistician, you would set up probably a lasso regression problem and hope that the lasso penalty zeroes out a lot of your coefficients. What we do is that we leverage some biologically motivated feature selection. And how many people here are familiar with gene ontology? Anybody? Okay, one person. Okay, a gene ontology is just a standardized vocabulary that describes how genes function within biological processes. So every gene is tagged with one or more of these gene ontology terms, and gene ontology terms, we have approximately 45,000 of them. And we've had this, this has been a decade-long running effort within the NIH to organize AD biology into what are known as AD biological domains. So so a biological domain is a specific aspect of AD biology, and there are about 19 of these biological domains. 3 of these are listed here. And so what happens is that when you take a biological domain, you can take the Go terms. The way the biological domains are set up is that you create clusters of Go terms. You take Go terms that are specifically relevant to any given biological domain. And all of these biological domains are mutually exclusive in terms of what Go terms are assigned to them. So you take ATP metabolism, for example, and it has a set of Go terms assigned to it. Those Go terms are not assigned to any other biological domain. Then you look at the Go terms that are assigned to ATP metabolism, and then you go into this list of 20,000 genes and you figure out which genes have annotations for the Go terms within that biological domain. So you go so when you do that sorting, you go from 20,000 genes to, for ATP metabolism, about 150 genes. And the sizes of these domains, in terms of the number of genes that are allocated to them, vary a lot. The largest one has about a few thousand genes. The smallest one, which is not mentioned here, is called space, and it has about 50 genes. So you get 19 of these with varying sizes. Another thing that we do is, we did this feature selection where we reduced the size of the number of features that we have. With 20,000 genes, we reduced it to a few hundred. Another thing that we do is that we can place some price. So so it is a tabular data, but biomolecules do not operate independently. They have relationships with each other. What we do is that we place a network structure on top of this the feature space. And the way we do that is that we query existing molecular databases like Git and Reactome. in the interest of time, I won't go into how this processing is done, but given a biological domain, we have a set of genes, we query existing molecular database, then we get a network topology that describes the functional relationship between those genes relevant to AD. So then, then we look at how we set up the model. So we have data for, let's say, we have data in the form of the gene expression or protein measurements. We have a network topology. So you have some node features. You have a network topology. What do you do if you want to fit a neural network? You use a graph neural network. Graph neural network leverages something called a graph convolution, which, if you're familiar with standard convolutions, it's essentially a generalization of standard convolutions. So if you think of standard convolutions, you're pooling information from a small local region, that's your kernel size. That's essentially what graph convolutions do. You pass messages between the nearest neighbors of your, or the neighbors of your nodes, right? And then you aggregate all of those messages through some sort of permutation invariant function like a sum or an averaging or something. So you set up a feature extraction, collect all of those feature extractors, put them through a transformer encoder to integrate all of them together. Why a transformer encoder? Transformers rely on the self-attention mechanism. Self-attention is permutation invariant. Self-attention relies on dynamically computing these pairwise correlations by design, and so it's a very useful model for integrating information from heterogeneous sources. So end to end, that's what the model looks like. You take data from different modalities, pass them through GNNs, get some features, pool them all together through a set transformer. That's it. We benchmark this to an existing method. The two columns, the two violin plots that you need to be looking at are the yellow and the purple, which are at the end. The one that we develop is represented by yellow. So in most cases, we have 16 different curated multi-omics data sets across 16. I think we do better than the existing benchmark in 13 of them. The violin plot is for 10 trials. Another well-known issue within multimodal data fusion is that you don't always do your multimodal model does not always necessarily do better than the best performing unimodal model. This is a well-known fact within multimodal data fusion. So if you were to do any kind of multimodal data fusion analysis, this is something that you would always have to check for. So you can see here that the multimodal model does better than either of the unimodal models in terms of predictive accuracy. This is held out predictive accuracy. Okay. So we build models that learn some useful signals. So what? So ultimately, we're interested in asking biological questions. For example, what kind of biomarkers are relevant to this disease? How do they interact with each other? How do biological domains interact with each other, and so on and so on. When we do this sort of exercise, the goal is ultimately to use the trained model as an in silico oracle to ask and answer these types of questions. So we use integrated gradients to do this biomarker identification analysis. These symbols might not make any sense to you, but these are essentially the top 20 key genes and proteins that we identified, and we identified some useful information. So first of all, did we actually learn biologically relevant signals? What we see from this table is, among the list of top 20 genes, is that 9 of them are already known therapeutic targets for AD, and 6 of them are matrizome proteins, and what that means is that these proteins function within the extracellular matrix between brain cells, and they're responsible for the structural stability of the amyloid beta plaques, which, if you remember, are one of the hallmark signatures of this disease. So we recover some of these matrizome proteins And basically what this tells us is that we do recover some biologically relevant signals. We recover 11 novel therapeutic targets, and one of them in particular is quite interesting to us, which is the one ranked number 14, IQPF3. This is a gene that has recent studies have shown that it's implicated in Alzheimer's, in that it plays a role in stabilizing synapses, the connections between neurons, which degrades when you have plaque or tau buildup. Okay. And we'll also look at interactions across biological domains. This is just so biological domains, again, if you remember, are those clusters of AD-relevant biology. One thing that is particularly interesting, so we look at, first of all, if you look at the most important biological domain is the big red circle with lip in it, and that's basically lipid metabolism. This is again, as in terms of validating what we have done, this it sort of it validates what we already know about AD biology. Given that amyloid beta production actually occurs in lipid membrane microdomains, I should say. One interesting finding that we had from here is this link between the lipid metabolism and the apoptosis. Apoptosis is a programmed cell death mechanism. Recent studies have come up with this mechanism called ferroptosis, which is an iron-mediated cell death mechanism in which lipids are involved. So the fact that this particular interaction pops up seems to be validating some recent observations from other research groups. Okay, so what happened here? It's a big multimodal data fusion, not a big. It's a multimodal data fusion problem in ML terms. Again, the computational challenges here are that this is a high-dimensional problem. Input-output map is complex and nonlinear. You have interactive effects that you need to capture, and there's also data missingness problems. So the way we go about the fusion problem is we do a feature selection, which is biologically motivated, not data-driven. It's not a principal component analysis or any other data dimensionality reduction problem method. Again, we leverage existing biological knowledge to set up a knowledge graph that tells us the relationship between our features that is relevant to the disease. Because we set up this graph, we're able to leverage the machinery of the message passing machinery of graph neural networks, and then we use a transformer to integrate all of that information. We again use standard attribution methods like integrated gradients and integrated Hessians to answer first of all to validate the fact that we are actually recovering biologically relevant signals, but also to make some discover new potential therapeutic targets for AD, and also to identify an interaction between biological domains, which is new information. Okay. So there are lots of we're working on several extensions of this particular line of research. Separate from the graph neural networks and the molecular data, we also do imaging work. We haven't yet fused those two modalities together, so that's something that we're still working on. We're also working on adding new molecular modalities within the ROSMAP study. We're also working on clinical research. So there's Model AD is another multi-institutional research consortium that is tasked with developing mouse models for Alzheimer's. And so taking the information that we get from molecular measurements in mice and translating that to the context of the human genome is a difficult translational problem, and so we're working. So that's another direction that we're working on, and that's basically it. And if you have any questions, please feel free to talk to me. Thank you.
For Q&A, so we have that for about 5 more minutes, and there's no mic right now, so if you have a question, you could just say it out loud.
I haven't really worked too much with graph neural networks.
Sorry, please.
I haven't really worked too much with graph neural networks, but I was wondering if you could just explain the difference between the input for a graph neural network versus the input for just a regular neural network and how that's different.
Okay. Well, so you know what a graph is?
Yes.
Basically. So a graph has nodes, it has edges, both nodes are connected by edges, and nodes will have attributes, right? So for example, you have a graph of genes, for example, or let's say you have a vector of gene expression measurements. It's 10, let's say it's 10 genes, you have some numbers, and that's just a vector. In a normal neural network, you would set up a multi-layer perceptron or something like that. Is that what you mean by like a regular neural network? Or?
Yeah.
Yeah, so something like that. You would just set up a multi-layer perceptron that takes a 10-dimensional input. You map it to whatever target you have.
Yeah.
With graph neural networks, you have this additional constraint that there is a network topology on the space of the input feature, so you have 10 input features, the measurements for those 10 features, but also those features are related to each other, and somehow that's determined by the topology of the network. So if gene, let's say gene 1 is related to gene 2 and 3, how can you make use of, how can you leverage the information from gene 2 and 3 and pass on that information to gene 1 because they're connected together. And that's what message passing really is. You're looking at a graph, you're looking at pairwise relationships, and whatever features have relationship with each other, you're trying to pass messages between them. It's like how, so that's essentially what a graph neural network tries to do. It's based on the fundamental machinery of graph neural networks is what is known as a graph convolution. It's exactly, or it's really just a generalization of, if you've done image problems, you might have set up convolutional neural networks. It's exactly that, except when you do convolutional neural networks for images, images are a regular grid, and it has a more straightforward graph structure. They also do the same thing. You look at patches of images, right? You look at patches of images, you compute some feature based on looking at a small patch, then you strike the filter, compute some more features, strike the filter, and so on. And you're looking at small patches. Same thing, except you're not looking at patches, you're looking at neighborhoods in a graph. Right? So that's determined by the message passing structure. That's all that the graph convolution tries to do. It's just a generalization of the standard convolution that you might have worked with.
Except there's some sort of hierarchy relationship.
Yes. Right. Yeah, it's not a grid, so you have to have some way of making sure that there's permutation in variance, right? So you can flip around the order. Like in an image, you can't flip around the order of pixels. That would not make any sense. Right? But in a graph, you can flip around there's no order on the set of nodes. So yeah.
I think basic question, but what's the reason for choosing a graph convolutional network over a graph retention?
A graph retention is also a graph convolution. Yes. That's a good question. Oh, sorry. But that's a good question. Yes, it's graph convolution is a general term for many different types of layers, I guess. Deep learning layers. So graph retention, like you just said, that's one type of graph convolution.
Thank you, Rohit,
Yeah.
for everything.